Clonal Compass is a single-cell immune repertoire copilot for researchers working with paired scRNA-seq and TCR-seq data. Researchers use this technique to connect what a T cell is doing with which clone it came from. This reveals whether the expanded T cell clones are associated with particular transcriptional states, tissues, etc. I built it because the landscape has a gap. Classical repertoire tools are strong at computation but do little interpretation. General single-cell AI assistants reason over gene expression but aren't repertoire aware. Clonal Compass sits in that open quadrant. It pairs clone expansion evidence with a Claude-powered interpretation layer while keeping the output grounded and reviewable. The app starts from computed evidence, not free-form guessing. It produces clone expansion metrics, marker evidence, and optional epitope lookups. In this demo, I included two sample reports, the PBMC baseline dataset, which shows low clonal expansion. In contrast, the T-cell infiltrating T-cell dataset shows a much stronger signal. That contrast helps demonstrate how the same workflow can summarize very different immune repertoire contexts. From here, we can ask questions of the data. First of all, which clusters show the strongest clonal expansion, and what would that imply? And here it answers straight away from the computed evidence with the most expanded clusters and largest clones. It's not guessing, it's reading back metrics. Next, we can ask, do any of the expanded clones match known epitopes, and what would that imply? And here we can see, this is where the tool's discipline really shows. The two most expanded clusters, the one with the biggest clones, returns no epitopes match at all. Instead of inventing a specificity to fill the gap or writing those clones off as meaningless, it states plainly that the antigen simply isn't cataloged in the database yet. Expansion without a hit means the specificity is unknown, not absent. The only expanded clone with a match are two cells within one cluster predicted influenza A, and it immediately flags that 2 cells out of 447 is far too sparse to call that population flu specific, and a predicted database match does not confirm reactivity. And then lastly, we can ask, does this data set indicate the donor was ill or had an abnormal immune response? And here we can see that it holds its diagnostic line and the data carries no clinical status or patient information. This is exploratory research analysis. And finally, the goal here is not to replace expert interpretation. The goal is to make the evidence trail easier to review so scientists can move from UMAPs and clonotype tables to a transparent, audible, first-pass interpretation.